interacts with other membrane proteins. These other proteins then activate a proton pump, right, which leads to cell wall loosening, or open some other ion channel in the membrane. Okay? Here's that second thing down here. Let's say that, and we would kind of get rid of this part here. If this was sort of right next to that, receptor changes shape, activates this B protein, which instead of opening up or activating a proton pump, opens up this calcium ion channel, and calcium ions come flooding into the cell. Okay, this is going to be very, very common. And we're going to pick up with this one here in just a second. Because if it opens up a calcium ion channel, then that's going to set off a whole other set of events. All right, so we'll pick up on that in a second. Okay. It turns out that calcium ions are of huge interest to the action of hormones. They seem to be working hand in hand with hormones to sort of get that final response from the cell. We'll see in a second that calcium ions are called second messengers. Right? The hormone is the first messenger, but the calcium ion is carrying on that original hormone message in the cell as a part of this series of biochemical events that are taking place. It turns out that calcium ion levels are usually very, very low in the cytoplasm. But the hormonal stimulation of calcium ion channels, in this mechanism that we just talked about, going from here over to here, allows for or results in the temporary elevation of calcium ion levels. So that elevation is most times under the control of a hormone message. So let's talk about what is going to happen next. All right. So we've got to the point of this calcium ion channel opening up. All right. A flood of calcium ions entering the cell. Those calcium ions are going to bind to calcium binding sites of certain proteins, right, within the cell. And the activity of those proteins is going to be altered, all right, in much the same way that the receptor protein was activated originally by the original hormone. So here we have a calcium ion doing much what the original hormone did, to that original receptor. So these calcium ions bind to these proteins and they alter the protein's activity. Now, there is a whole class of proteins called protein kinases that are a type of enzyme that is triggered or activated by calcium ions or other second messengers. Calcium ions aren't the only second messengers, but they're a very common one. So more specifically then, calcium ions are going to bind to calcium binding sites on protein kinase molecules. All right? And then those protein kinase molecules are going to modify other target proteins. This is a long chain of events that began with the hormone binding to the receptor. So these protein kinases are going to modify other target proteins, and many times what they do is they're phosphorylating these other target proteins. In fact, that's what protein kinases classically do. All right, they phosphorylate other proteins. And you probably know that when I say phosphorylate, they're transferring a phosphate group onto these other proteins. Okay, so this target protein now is phosphorylated, and the phosphorylation of that target protein will alter its structure and function, and it will go do something else. And many times in this sort of longer biochemical pathway, it's this target protein then that behaves as the transcription factor and goes to turn a gene on or off. Okay, so we've kind of talked about we've talked about pathways of three different lengths. There's a very short hormone pathway where the hormone binds to a receptor, boom, and they go directly and turn a gene on or off. we talked about this medium-sized pathway here with the cell wall loosening, where there's hormone to receptor, change the shape of a membrane-bound protein, which then opens up a proton pump. All right? And now we're talking about this long pathway where hormone binds to receptor, receptor interacts with this other membrane protein, which opens up a calcium ion channel. Calcium ions flood the cell. They bind to transcription factors, or they bind to protein kinase molecules, right? Those protein kinase molecules phosphorylate target proteins, some little phosphate groups there, and then that phosphorylated target protein then behaves as a transcription factor, turning this gene on or off. So that's sort of this top part here. Okay, so that's the longer pathway. That's very common though. It's going to be that long between the original hormone message and this gene expression. It's going to be that long. All right. How did I just go backwards? Oh yeah, I can go backwards. And so I've already said this, but calcium is mediating hormonal responses, and so it's called a second messenger. It's a second messenger because it's transferring information from the hormone receptor complex to the target protein. And by doing this, it's amplifying the hormone signal. Okay, this is very, very important. This goes back to something I said on day one of hormones, is that you don't need a whole lot of hormone to get the desired effect, the intended effect. And part of this is because there's signal amplification at several steps of this biochemical, during several steps of this biochemical pathway. So for example, this is a very simple example of what I'm talking about. And I'm probably underestimating the amplification here. So one hormone molecule binds to one receptor protein, which opens one ion channel. But from that, thousands upon thousands of calcium ions flood into the cytoplasm. Each one of those calcium ions will activate a protein kinase molecule. Each of those protein kinase molecules will phosphorylate many target proteins. And each one of those target proteins will act as a transcription factor at least once, if not more than once. So I'm just saying that there are 2 points of amplification here, but there are probably even more points down here at the level of what the target protein then ends up doing. Okay? So this, on a molecular level, helps to explain why you only need a tiny bit of hormone to get sort of this huge cellular response, because there are these points of amplification in these biochemical pathways. Okay. Let's see. Yeah, let's talk about, before we go into the morphogenesis thing, let's give you one example from the book of a hormone response pathway. All right. Now look, this has nothing to do with, this isn't going to be, we're not going to turn on any genes here, so we're going to kind of back off of that. That's a major thing that hormones do. They do lead to gene expression. But here's another example. This is a good picture from our book. You remember me saying that abscisic acid causes stomata to close. All right, but why is that? Okay, so here's why. Look, here is a hormone receptor, an abscisic acid receptor. Okay, here's abscisic acid plugging into that receptor, activating it. That leads to the opening of this calcium ion channel. All right, so calcium ions come flooding into the cell. All right, so here we have, so here's the calcium ion. It's going to bind to this protein kinase molecule. Right, now at this point, the chloride ion channel is closed, and so is the potassium ion channel. But here we have a calcium ion that's going to plug into this protein kinase molecule. So let's jump over here. that protein kinase molecule leads to the opening of this anion channel, and both chloride and malate ions are going to leave the cell. All right? That's going to depolarize the membrane, which will open up the cation channel, and potassium ions will leave the cell as well. And when you look up here, if potassium ions and chloride and malate ions have left the cell, those guard cells, remember you got a stomata, but you got the guard cells. The guard cells become flaccid, and the stoma closes. All right, so here we have a hormone message, and a second messenger leading to the opening of these two sets of ion channels. All these ions leave the cell, those guard cells, which then become flaccid, and then they close. Over here, when all of these ions are inside the guard cell is turgid. It maintains that semicircular shape, and so there's a hole in the middle of it. But if it kind of relaxes, then the stoma closes. All right, so this is just another particular example of what these hormones are mediating or leading to. Okay, so you got the proton pump thing. All right, the transcription factor thing, either directly or indirectly, and then here a molecular mechanism for stomatal closing. Okay, so let's switch gears now and talk about the morphogenesis thing. Right? How is it that on a molecular level, on a cellular level, hormones are influencing the overall shape of plants, right? The overall, the morphogenesis of plants. I told you that morphogenesis depends on the rate of cell expansion, right? The direction of cell expansion. So let's kind of take it from that angle. Okay. So the rate at which an individual cell expands is controlled by turgor pressure, right? Water pressure inside the cell. All right. There's a whole bunch of pressure inside the cell that can cause it to expand quickly. Not so much pressure, expand more slowly or not at all. But the rate of cell expansion is also determined by the extensibility of the cell wall. Right? You can increase the rate of cell expansion by having an increased pressure, or you can kind of loosen up the cell wall. And then the same amount of pressure will cause it to expand more quickly. So we can make the cell wall more extensible, we can loosen it up. So the extensibility then is a measure of how much the wall will stretch permanently when a force is applied. So here's a good picture of this. Rate of cell expansion depends on turgor pressure and cell wall extensibility. Hormones have very, very little, if any, influence on turgor pressure. So that's not where they're exerting their influence. But they have a huge influence on the extensibility of the cell wall. And so hormones can thereby influence the rate of cell expansion. So check this out. One of the reasons why the auxins and the gibberellic acids stimulate plant growth, right? We said that in the last two lectures. Auxins and gibberellic acids stimulate plant growth because they can increase the extensibility of the cell wall. They can loosen it up so that the cell walls expand more rapidly. Abscisic acid and ethylene, on the other hand, we said, inhibit plant growth, and that's because they decrease cell wall extensibility. All right, but so how exactly does this happen? All right, so there's a couple of hypotheses. The acid growth hypothesis is the one that I kind of gave as a general example, that hormones such as auxins are leading to the activation of a proton pumping enzyme in the plasma membrane. I showed you that in a little picture. Protons or hydrogen ions are pumped into the cell wall, and that the drop in pH loosens that cell wall structure. All right, so that's kind of what was modeled for us right here. Okay, so this mechanism right here. here to here to here. That's the asset growth hypothesis. All right. There's an alternative hypothesis which turns out to not be that much different.